The two questions in particular, two answers were accepted for the quiz. But the answers that are on the quiz are the questions you need to think about going forward. And the whole idea revolves around the idea of activator proteins. Sometimes have activating molecules, sometimes they don't. So for that situation, we do have proteins that can bind DNA by themselves, or do require a small molecule to bind before they can bind the DNA. All revolves on that idea that we talked about. I know I mentioned it in lecture, and then not a whole lot of people remembered that particular part. So the slides have been changed and updated, but you'll need to make a note. Inactivating proteins don't need a small molecule all the time. Sometimes they do, sometimes they don't. It's all sort of dependent on the type of activator protein. So on this question here is actually worth 2 points, which is why there were 2 answers. I don't know if anybody ever looks at the point total to guess how many answers they need to give. It's not always a bad option, so look how many points there are to guess how many answers it might need to be. But look at this question, it says bind DNA without a small molecule. And that's the activated protein can do it. It doesn't require a small molecule, but it can bind a small molecule to bind DNA. And then the repressor by itself does bind DNA, and when it binds a small molecule, it releases DNA. So that's one thing you want to remember. There's some confusion with what proteins do once they bind to the small molecules. So make sure you review that for the exam. It's very easy to get confused because of the proteins doing sort of different things. And again, for this question, there were only one answer I was looking for if you take into account that activated proteins don't need a small molecule all the time. So the aporepressor would be the only protein that does require a small molecule all the time to bind DNA. Activators do sometimes, but not always. But because there are different possibilities, if you follow the notes to 100% in what they say, then you should have answered aporepressor and activator. So here, if you just answered aporepressor, that's 1 point. If you answered both these, you also got 1 point. On this one, if you answered both activator and repressor, you got 2 points. If you just answered repressor and nothing else, you got 2 points. If you answered repressor and something else, probably got 1 point, because that other answer was an incorrect answer. And I remember if you got 1 point if you said activator and something else. I don't remember what I gave you, but take a look at it. If you have a question about your point total, let me know. I'll double check things. Make sure you're getting credit for things you should be getting credit for. The other thing you may see in the quiz is formatting for enzyme names and protein names is important. They're written in certain ways for certain reasons, so please make sure you do format them the way they are formatted. Like Cas9, the C is capital, the AS is lowercase. That's an important thing for the organization. For RAD, all 3 letters are capitalized. For RET-K, it's only R capitalized and E and C in lowercase and A is uppercase. Writing these in the wrong format is going to mean something different. Please make sure you do write those and study those in particular orientations or formats. It basically comes down to how we name proteins in eukaryotic organisms and how we name proteins in bacterial organisms. For bacteria, the first letter is always capitalized. For eukaryotic organisms, all 3 letters are capitalized. Typically, we also see numbers with eukaryotic proteins rather than letters, and bacteria use letters rather than numbers. But you don't need to memorize that, but I do want to make sure that you write these things down correctly. It didn't affect everybody, but there were a few people that unfortunately got, or Cas9 as all lowercase, and that's not correct. It'll also be true as we talk about other proteins today, so we got to focus in on which letters are capitalized and which ones are not, because it will be important to think about. Does anybody remember what we talked about on Friday? We talked about this big circle thing. Put it over here. The cell cycle, interphase being G1, S phase, and G2. The M phase being mitosis and cytokinesis. S phase is when DNA replication occurs, which is our really first important step before cell division. And then G1, G2 are thought of as this recovery and preparation periods. G1 is getting ready for replication, G2 is getting ready for mitosis. And as we think about regulating the cell cycle, we want to highly control when certain things happen because we're trying to control when we copy our DNA, when we don't copy our DNA. Again, we should limit. DNA copying should only happen during the S phase, not during G1, not during G2, and definitely not during the M phase. So how do we control these types of things? We'll get into that in a second. But we also talked about there are some important checkpoints. One checkpoint is right here. This is one of the more important checkpoints to make sure the cell is ready to go through cell division. At this point, nothing bad has happened, so we can stop and pause everything and wait until we're ready to do it, or fix everything that might be broken for that point. The other important checkpoint is right here near the end of G2. Here we're doing a check for DNA, but we're checking also to see if our DNA is sufficiently broken. I mean, we want to just go the route of apoptosis rather than make two cells that are going to be crappy. Now, it's not specifically a checkpoint, so I'm using a different color, but there is another little thing going on in the middle of mitosis that we'll talk about. It is an important sort of regulatory step. I call it an event. We'll continue to call it an event because it's not a checkpoint, but in the truest sense of the word, checkpoints. What controls the cell's progression through the cell cycle from G1 to S phase, from S to G2, from G2 to M phase, and even throughout some of the things that go on during mitosis? We use protein that is made up of two different components. Are called cyclin kinases. And cyclin kinases are involved in allowing the cell to move through the various stages of the cell cycle. The cyclin protein is what it sounds like. It cycles. Its expression patterns will vary. And as it gets produced, activity happens. When it's not produced, activity does not happen. The kinase protein is what's called a phosphorylating enzyme, meaning it adds phosphate groups to other proteins. The kinase does not function when its cognate cyclin protein is absent. So the cell can continually make the kinase protein, but it doesn't do anything unless it's also making the cyclin that goes with it at the right time. So these things are called cyclins because their expression does cycle, like the cell cycle. So they'll turn on at some point, the kinase becomes active, they'll turn off, the kinase becomes inactive. We cycle between inactive and active. It might be a weird way to go about it, but you might think, why not just regulate the kinase protein's expression? But that's not what the cell cycle does. The cell cycle regulates the cyclin proteins rather than the kinase proteins. There's only one real explanation for this that I could give you, is some kinase proteins work with other cyclin proteins. Sometimes you need a kinase protein in other places than just in one place. So kinase proteins are going to phosphorylate, meaning we add a phosphate group to a protein. And when we phosphorylate, we're going to change activity. It's important to pay attention to what sort of activity is going to go on as we change these things. Most oftentimes, for what we talk about, phosphorylating proteins is going to turn them off. So they're going to lose their activity. When they're unphosphorylated, they're active. When they're phosphorylated, they're inactive. We won't talk about it specifically for us, but anytime you phosphorylate a protein or phosphorylate anything, there's also sometimes a need to dephosphorylate that thing. In this case, we use an enzyme called a phosphatase. As I say, don't choose a messenger. I did not name these things. Why is the phosphatase the thing that removes the phosphate rather than add it? I don't know. Ask a chemist why it's a synthase and not a synthetase. Why aren't those two the same things? Weird stuff. Anyways, kinase is what we care about. Kinases phosphorylate proteins, and protein phosphorylation is going to inactivate the thing rather than activate it, or turn things off by phosphorylation. We have a few different cyclin kinase protein pairs to deal with. There are 3 that are very important for us, and some that are not important for us. As you can see, the 3 that we care about most are called the CDK4 cyclin D pair, CDK2 cyclin A, and then CDC2 cyclin B. Again, don't shoot the messenger, but why did we change the format at the end and go from CDK to CDC? Other than just to mess with everybody and make it harder on people, I have no idea why. It can't be CDK and some other number. We got some weird things to have to deal with. And it's one of the things you'll just have to worry about as you study this stuff. 2 goes with B and 2 goes with A, but K goes with A and C goes with B, and it'll get weird after a while. And our main focus is going to be right around here between the G1 and the S phase transition, which is going to involve some of these cyclin kinase pairs, but not all of them. And in particular, it's going to be the first one that we're going to talk about the most, and the most often, and the first, the CDK4 cyclin D. And we'll pick these up as we go around. But this is not a picture to memorize, it's just an illustration for some of the differences that we see in expression patterns. But we'll pick these up as we go along and see sort of basically each one of these checkpoints is each one of these cyclin kinase pairs, or at least somewhat related to these cyclin kinase pairs. Because one is actually not listed on here, but we'll add it as we go around. So what we need to remember also from last week is this idea of what do we want to do. As we transition from a G1 to S phase, we need to start something. And so somewhere, either near the end of G1 or near the beginning of S phase, we need to start the expression of our DNA replication genes. Everything that is needed to copy DNA has to be made before we enter S phase, or immediately upon entering S phase. We need to have all those enzymes and proteins ready to start copying our DNA. You can think of this as the sort of the start of S phase, because once those things get made, DNA replication starts. So near the end, near the beginning, it's not that important to sort of specify where it's actually happening. At some point or another, we're going to start this process, and that is basically when S phase is going to begin. And also, near the end, we need to do the opposite. And now that I think about it, I should have used different colors. But again, near the end, we need to do the opposite, so we need to stop expression of DNA replication genes. Because we only want to replicate our DNA during the S phase and never during any other phases of the cell cycle. So turn them on, and then eventually turn them off. So everything we do is going to facilitate this turning on process and then turning it back off. There's one other sort of weird thing about the naming system in eukaryotic organisms. A lot of the genes are not name for what they do, but for the disease associated with that gene being mutated or wrong. And so we first have a gene called RB. Not roast beef, but RB. Not RBs, RB. But think roast beef, or think chicken, or think mozzarella sticks and curly fries, apple turnovers. Does anyone like Arby's? Nobody really likes Arby's? Okay, RB is a gene. It's also a protein. It's also an abbreviation for a disease called retinoblastoma. You can probably guess what retinoblastoma causes. Eye cancer. Not exactly eye cancer, but any type of tumor in the retina. Now it turns out that if you mutate or do something to the RB gene, that's what causes uncontrolled cell growth. And so that's where we get these things, is you identify a disease and then that disease becomes the name of the enzyme or protein that's affected, rather than what it actually do. So RB does not cause eye cancer, it does something else. And in particular, the RB protein is going to be most important in keeping the cell trapped in G1 phase. So that is its whole role. Keep the cell in G1. Which means while RB is active, the cell can't progress through the cell cycle. That's our starting point. RB protein is a protein that is important in keeping the cell in G1 until the point that we're ready to move on to the next phase. There's another protein or another gene that we call E2F. E2F is a transcription factor that will turn on and express all of the replication genes. So this is a regulator, and the regulator controls replication. We call it a transcription factor instead of a regulator because it does sort of the same things. It is something that is going to be needed to transcribe all the genes that encode gene replication enzymes. One protein that wants to keep the cell in G1, another protein that is needed once we get into S phase. That's right now what we need to sort of understand. E2F is needed for S phase. Okay, so E2F makes S phase happen. RB keeps S phase from happening. E2F makes S phase happen. Which means there is a competition of sorts that's going to go on between the RB protein and the E2F protein. This competition is going to control when we progress, or if we progress, to the next phase of the cell cycle. We'll talk about how this happens as we go along. We've got the RB protein, again, what it does. The E2F protein, what it does. We also have the cyclin kinase pair. CDK4, cyclin D. This is the cyclin kinase pair that will phosphorylate the RB protein specifically. So now we have a third sort of player in this thing. CDK4, cyclin D. RB, E2F. These three things are the first bits of information we need to sort of understand how we progress through the cell cycle. And so let's just look, as we progress through the cell cycle, what do we have? At the bottom is going to be a sort of running total of what's going on, but if we're in G1 phase, the RB protein is active and the E2F protein is inactive. And what is important here is, why is E2F inactive? That's because of what's called a protein sequestration process. The RB protein binds to the E2F protein, which prevents it from doing anything. Whatever you want to think about, tackle somebody. Throw them in a dungeon if you want to get a cob. a bag, I don't know, lock them up. Whatever you want to think about, RB protein is keeping E2F from doing anything by just physically holding onto it. And RB is active because the kinase protein that phosphorylates it is not active at this point. So we start out this process, the RB protein is active, it binds to E2F, which keeps E2F inactive. We're going to see a change or a switch occur as we get closer to what we need, when we need E2F. So as we go closer to S phase, we need to be able to make a switch. This is where we start with RB active, E2F inactive, CDK4, cyclin D also inactive. Might be better to write it down at the bottom also about the kinase proteins, but my little legend at the bottom is just tracking E2F and RB. So as we get closer to S phase, we get closer to the point where we need to start thinking about doing something different. We need to change. And if everything is working correctly, the cell is right. And as we're going to see as we go forward, right means is our DNA undamaged? Is our DNA ready to be copied? Is the cell sort of undamaged also? As long as the cell and the DNA are in good standing, then once we progress through the cell cycle. If there's everything wrong with the cell or wrong with the DNA, that's typically a trigger to stop or pause the cell cycle and not continue until things are fixed. We don't want to make bad cells, because bad cells are just going to make bad functions and possibly lead to diseases. So as we know that the cell is ready and primed and all correct, we can move from G1 phase and move into the S phase. And to do this, we need to inactivate the RB protein. And that is going to happen by phosphorylating RB. So now RB becomes inactive because of phosphorylation. The CDK4 cyclin D is the thing that phosphorylates the RB protein. Once we phosphorylate RB, it can no longer bind E2F. So we free E2F by phosphorylation of the RB protein, which allows E2F to go bind DNA. Once it binds DNA, it does what it does, which is to make lots of things. In particular, the genes that are required for replication. So we start off with RB active, bound to E2F, so E2F is inactive. We phosphorylate RB, E2F is released, becomes inactive, and it transcribes all the genes we need for replication. So RB is in the inactive state because it's phosphorylated. E2F is in the active state because it's now released from being tightly bound by another protein. One of the other things that's kind of neat about this process is E2F will also express the other cyclin kinase pair that becomes important, CDK2 cyclin A. So RB is phosphorylated by CDK4, cyclin D. This other cyclin kinase pair is going to phosphorylate something else. So now we've accomplished the first step. We need to start the expression of denervation genes so that we can enter S phase. At the end, we need to stop the expression of denervation genes. So what are our possibilities of things we could do? So first, what do we need to do? What's the sort of general thing that we would need to do to stop expression of denervation genes at this point? What did we just do to make?
Inactivate E2F.
So that's what we need to accomplish in Sabrina, to activate E2F. And how could we do that? If you think of what ways could we do to go about inactivating E2F?
Activate.
And one way would be very easy, just to dephosphorylate RB, so it binds to E2F, and we get back to the reset state. Unfortunately, that's not what happens. RB has to stay inactive for a longer period of time. We don't want RB to become active during G2 phase, because RB keeps the cell in G1 phase. If RB becomes active, then we basically sort of go backwards and might go through replication again without doing cell division. We have to keep RB inactive. That would be an easy thing to do. We can't do it. So we're going to leave RB inactive. We have to do something else to inactivate E2F. And that's where the cyclin kinase pair comes into play. It is the thing that will actually inactivate the E2F protein. So instead of dephosphorylating RB, we leave RB phosphorylated and phosphorylate E2F instead. So we no longer transcribe those replication genes. And everything sort of should go back to the next phase. So this interplay of phosphorylate one thing, phosphorylate another, controls our transition from one phase to the next, and from one next phase to the third phase. So again, if you want to put a little note here, CDK4 cyclin D makes this transition happen between G1 and S phase. The CDK2 cyclin A reaction makes this transition happen between S phase and G2 phase. And hopefully it makes sense. We don't want RB to be active yet because we're not entering G1 yet. It's not until we get near the end of M phase that we would want RB to be active again to signify that now we're in G1 again. And eventually we're going to have to dephosphorylate E2F to reset the system. But that's not something we care about. We just care about how do we deal with E2F during G1 phase, and how do we deal with E2F during G2 phase. During G1, RB binds E2F, keeps it inactive. During G2 phase, E2F is phosphorylated, which keeps it inactive. If you approach it like that, it's sometimes a little easier to sort of think about, well, we need to remember, why is E2F inactive during G1 phase is different than why E2F is active during G2 phase. So is this too complicated? We've got RB, E2F, CDK4, cyclin D, CDK2, cyclin A. It's only 4 proteins. Is it that complicated? We can make it more complicated. In fact, we are going to make it more complicated. Just slightly. But this is, again, sort of the important part is RB is what keeps the cell in G1, so as long as RB is active, we're stuck in G1 phase. E2F is what's needed for S phase to happen. Neither RB nor E2F are needed for G2 phase to happen. So you can sort of switch what's going on as we go along. But ultimately, it's not RB, E2F, and the cyclin kinases that control this process. It's a protein that's called a P53. And P53 plays a very important role in controlling when does the cell undergo cell division. You see, it's a tumor suppressor gene. So let's revisit, what does tumor suppressor gene mean?
Turns it off.
Turns what off? When we talk about tumor suppressor genes and proto-oncogenes, there were 2 aspects we looked at: the cell division and apoptosis. And those 2 things were opposite for proto-oncogene, and then switched for the tumor suppressor gene. So either one is promoted and the other is inhibited, and then we reverse it for the other group of genes. And so if we're trying to suppress a tumor, what are we trying to do?
Increase apoptosis and inhibit cell.
We can worry about the cell division first, but I think it's a little more important. One of the things we're going to talk about and continue to talk about is tumors have uncontrolled cell growth. So if we're trying to prevent something from having uncontrolled cell growth, we're trying to limit cell division. And also, if something has uncontrolled cell growth, we're also trying to get that cell to sacrifice itself. So we're trying to turn down expression or turn down replication or division while increasing or promoting apoptosis. Tumor suppressor genes are trying to stop cell division. So the ultimate point of this is tumor suppressor genes like P53 are trying to stop cell division from actually occurring. Which means things like proto-oncogenes are trying to make cell division actually happen. As we go a little bit longer, or a few more slides forward, RB protein and CDK4 cyclin D will fall into the category of being either a tumor suppressor gene or a proto-oncogene. But E2F is neither one of those two, it's just a different category of things. You see that P53 does control a lot of things, but one thing that is important is that P53 is controlled by a protein called MDM2. I have no sort of tricks here. MDM2 goes with P53. And so sometimes P53 is on, sometimes P53 is off, and that's going to affect our ability to transition through the cell cycle properly from G1 to S phase, from S phase to G2, and further on through the things. And one of the other reasons to think about P53 is the fact that in a lot of different types of cancers, P53 is mutated and malfunctioning. It's not every single cancer, but in a lot of cancers, it is a problem. So it's a pretty important player that we can deal with. So when we lose P53 activity, we lose a few different things. Remember, P53 is trying to prevent cell division, right? P53 is trying to prevent cell division. Does that mean we stay in G1 phase or move to S phase? Yes, people want to stay in G1 phase. And if you lose P53, you lose the ability to sort of stay in G1, so you start cell division when you're not supposed to start cell division. And the other thing, because tumor suppressor genes try to promote apoptosis, losing P53 function means this no longer happens, so apoptosis is less likely to occur. And so we're more likely to go through cell division and less likely to die when we lose any tumor suppressor gene, especially P53. Which means the opposite would happen if a tumor suppressor gene, a proto-oncogene was lost. Just to give you some stats, there was a paper that can't do this in humans, so they did it in mice, where they looked at doing what's called a gene knockout. And so we have two different types of mutants. One where we have a heterozygous situation of a good copy of P53 and a mutated copy. And then the homozygous recessive will be the last one to column here, where both P53 copies have been inactivated. And what you can see is everything sort of goes to shit really quickly when you lose one copy of P53. Your chance of getting a tumor is now 1 in 2 rather than 0. Your life expectancy is half. And so, yeah, losing one of these is a bad thing for the cell because things get worse. If you lose both of them, you're not completely sunk, but instead of living 120 weeks, you lose 20. So live it up in those short weeks. That means like 1 through 5 is your adolescent period, then 5 through 10 is your teenage years, and then 10 through 15 is your adult years, and then 5 through 20 is your elderly years. I don't know. Imagine only living 20 weeks. Oh, sorry. I guess it's better than 1 day for some insects. But P53 is pretty important. P53. Pretty important. More important than P90X, whatever that is. One of those P90Xs. Those built muscle. Anyways, because P53's importance in the overall health of the organisms, gene therapy is targeting P53 a lot. So can we replace P53? That will help, hopefully, in a lot of different cases. Because most times it's broken. If we can fix it, that'll help somewhat. P53 responds to a specific occurrence, and that is DNA damage. So anything else could go wrong, and P53 will stay inactive, but as soon as DNA damage is detected, P53 becomes activated, which means we stop where we are. And hopefully this is what happens during G1 phase. Before we even copy our DNA, We check to make sure there's no mistakes, no damage, no errors. If there is an error detected, that's why we pause during G1 and enter that G0 phase. Try to fix the things. And if we can't fix them, then let's undergo apoptosis, because this is an unrepairable piece of damage. If things can't be fixed, they get fixed and everything goes back to normal. But this DNA damage could occur during G2 phase, in which case the cell could say, things are wrong, we need to stop progressing through the cell cycle until we fix things. If things can't be fixed, then apoptosis might be able to happen. You can see here there's two transitions that we're worried about. Transitioning from G1 S phase, but also transitioning from G2 to M phase. And stopping these stops the cell cycle from continuing. So it's not just that first checkpoint, it's both checkpoints we can sort of deal with. Is DNA damage? If so, we can stop here and try to repair some of the problems. And these are the two most important things. P53 is going to limit or make these things possible by preventing the cell from keeping through the cell cycle. There's also something that happens during S phase, but we don't really talk about this a whole lot for replication, so it's not that important to memorize. But if you care about it, you can read about it or you can ask about it later. But P53's function in keeping the cell in G1 phase or keeping the cell in G2 phase are the two most important things. And that sort of controls the cell division aspect of tumor suppressor genes. But also, if damage is not repairable, P53 will play a role in initiating apoptosis. So P53 is able to do both things. It will try to fix things first, and when things can't be fixed, throw it away and start over. So one of the things you need to think about again is, what does DNA damage cause? It causes this transition for what we want to happen. When DNA is undamaged, we go through the cell cycle as it is without any pauses or alterations. But as soon as DNA damage is detected, we basically stop what we're doing until we can fix it. If we can't fix it, apoptosis is the alternative. Again, P53 is going to be involved in both these two things: controlling cell division and controlling apoptosis. Beyond that, there is no other crossover between the things that are involved in apoptosis and the things that are involved in controlling cell division. And logically, it makes sense. If DNA is undamaged, keep copying DNA and keep making new cells, because everything is normal. Everything is healthy, everything is good. Or, everything is awesome. Everything is cool when you're part of the team. Everything is awesome. Now our team's gonna get bigger, but only bigger by one. There's only one new protein on this slide we haven't heard about before, and that's P21. This is what I call an interaction matrix, because it tells us which proteins are interacting with other proteins. And what we're going to do is we go forward to see, are these on or off, what happens relative to what they do. They're also color-coded, so we can remember, hopefully, that P53, P21, and the RB protein are considered tumor suppressor genes because they want to inhibit replication or cell division, which is what we care about here. Actually, we can ignore apoptosis because apoptosis is not any of these things. Actually, better. Let's get rid of that. Get rid of that, because who cares right now? Those are not really things we worry about. Tumor suppression genes will stop replication, meaning we stop cell division. Proto-oncogenes, which in this case there's only two of them, the MDM2 protein and the CDK4 cyclin D, will promote cell division. Remember, what does CDK4 cyclin D do? Phosphorylates the RB protein, which releases the E2F protein, which starts S phase. So these two things are competing. If this is active, this is inactive. If this is active, this is inactive. That's one of the things we'll see as we go along, is this sort of cycles. As we go backwards, we're gonna see active and inactive things go along. And rather than look here, we have all this in these other slides, so might as well use them. But as you see at the top, when DNA is undamaged, we are going to progress through the cell cycle normally. And so if DNA is undamaged, we want to go from the G1 phase to the S phase. And that requires E2F to be active. This is what is needed to make S phase happen. E2F needs to be active. For E2F to be active, the RB protein has to be inactive. The reason why the RB protein is inactive is because the CDK4 cyclin D phosphorylated it, which makes this protein active. The reason why this protein is active is because this protein is inactive, and this protein is inactive because this protein is inactive. Is what this is trying to tell you here. This step doesn't occur, this step doesn't occur, and this step doesn't occur, because all things are going on. The red arrows and the green arrows are supposed to tell you if this is a good thing or a bad thing. Is this a positive step or a negative step? Is it an activation step or an activation step? So remember, MDM2 keeps P53 inactive. Because P53 is inactive, P21 is inactive, which makes the CDK4 cyclin D active, which phosphorylates RB, turning it off. RB releases E2F, E2F binds DNA, and we get replication and S phase occurring. So it's just a bunch of toggle switches. How many toggle switches are there? 1, 2, 3, 4, 5 toggle switches? Up or down is what we want to think about. Again, there's two ways to approach it. Start at the end and work way backwards. Because this is the part of the story that we should have the most information about, and we talk about the most. And this is sort of the link between what we talked about first and what we're talking about sort of now. Is P53. What does P53 do? This is only as we go from G1 phase to S phase, meaning that late G1 phase, early S phase is when these events are happening. It's not about what goes on early in G1. It's not about what happens late in S phase. It's just that first little bit between G1 phase, S phase transition. These are the things that need to happen to allow the cell to continue with the cell cycle and make replication happen. If you learn this really well, then the next thing to think about is very useful, because you're just turning things opposite. And what you'll notice is everything that's red here will either be stopped or a different color going forward, and vice versa. Because in this next slide, we're looking at what happens if DNA is damaged, meaning we want to stop the cell cycle. So we're at the very end of G1, we're going to pause in G1 phase and not transition into S phase. And so for that to happen, RB needs to be active, so it binds D2F, and we stay in the G1 phase. For RB to be active, this has to be inactive. And that's what the P21 protein does. P21 will inactivate the CDK4 cyclin D. And the reason that P21 is able to inactivate our cyclin kinase pair is because P53 is now active. And so something happens when DNA damage is detected to get rid of MDM2. I don't know what the answer is. You don't need to know what the answer is. Just something happens where P53 is no longer inactive anymore. When P53 activates, it activates P21. P21 turns off the kinase. So RB never gets phosphorylated. RB stays active. RB keeps the cell locked in G1 phase. Until things are fixed, we can reset the system and allow everything to continue forward. And the whole story is about what are RB and what are E2F doing. Or really, what is the RB protein doing? Because RB is a really important control point of this whole thing. What is happening to RB and what is RB doing? Okay, when DNA is damaged, we need to stop the cell cycle. So we stay locked in G1 phase. When DNA is undamaged, we want to move from G1 phase to S phase. So whatever that takes to happen, well, make that happen. Turns out that P53 does a whole lot of stuff. Not that we need to look at all these different things, but it is a pretty important part of this whole thing. And even for what we talk about, there aren't as many things that we talk about that are actually known. So we're on this side of the story right now with RB, E2F, CDK4 cyclin D, P21, P53, and the MDM2 protein. We're not gonna talk about this or that, or this down here at all. But here's an miRNA. What are miRNAs involved in doing? The first thing you should think about is meh.
It has like many targets.
It controls.
Many targets
other
of
RNA,
what?
right? It's other RNA.
There's a phrase we're missing. Like the name of the whole thing is up here, and siRNAs and miRNAs are the players, which can control translation by degrading mRNAs. But what is this whole process called? Is the thing I want you to think about. RNA interference is the answer. Just the I is actually the inhibitor thing. But I want you to just understand that this is a lot more complicated than we can make it. I'm trying to make it less complicated. Have I succeeded? We'll see as we go forward. But on Wednesday, we're going to start with this slide on the board. We're going to do all things out.